A good hypothesis is essential to figuring out the source of illnesses during a foodborne outbreak investigation. In this video, we review the importance of hypothesis generation and talk about the 3 key pieces of information that investigators use to develop a hypothesis: known sources of the pathogen causing illness, descriptive data including person, place, and time characteristics of ill persons, and case exposure assessments or case interviews. Hypothesis generation is an important step in any outbreak investigation. In a foodborne illness outbreak, the hypothesis states which food vehicle or vehicles may be the source of the outbreak. Foodborne pathogens can be found in a variety of reservoirs and may be transmitted in several ways, making it difficult to identify specific food vehicles. This is especially true in clusters detected through pathogen-specific surveillance, where cases are geographically dispersed and don't have an obvious epidemiological link. A good hypothesis allows investigators to focus an investigation, conserve resources, and increase the likelihood of rapidly and conclusively implicating the contaminated food vehicle. Hypothesis generation is dynamic and iterative. It begins in the earliest stages of an investigation and is refined as more information becomes available. As information is gathered, a pattern or signal may emerge that investigators can begin to evaluate. It is important to involve a multidisciplinary outbreak investigation team in hypothesis generation, including experienced colleagues who can provide historical context. When generating a hypothesis, investigators should consider 3 pieces of information. The first is known sources of the pathogen causing illness, including reservoirs, sources implicated in past outbreaks, findings from case control studies of sporadic illnesses, and subtyping information. The reservoir of the infectious agent can indicate potential sources and contributing factors. Pathogens with a human reservoir, like norovirus or hepatitis A virus, are commonly associated with infected food handlers or ready-to-eat foods that have been contaminated with human feces. Pathogens with animal reservoirs, like salmonella, are commonly associated with food sources of animal origin or foods that have been contaminated by animal feces during production. Whereas pathogens with environmental reservoirs, like listeria, are commonly associated with foods that can become contaminated by soil or water. Past outbreaks can suggest common food pathogen pairs. For example, the foods most often associated with salmonella illnesses are seeded vegetables, such as tomatoes and cucumbers, chicken, pork, and fruit. Leafy greens and beef have been associated with E. coli 0157 illnesses, and dairy products and fruits have been associated with listeria illnesses. Subtyping information can help provide more detail, especially for pathogens that have multiple reservoirs. For example, Salmonella serotype enteritidis has been associated with eggs, whereas serotypes Uganda and Infantis have been associated with pork. The second piece of information that investigators need for hypothesis generation is descriptive data, the person, place, and time characteristics of ill persons. Person characteristics may include gender, age, race, and ethnicity. For example, dairy and animal contact outbreaks are more likely to involve young children, and women report consuming more fruits and vegetables than men. Geographical mapping of cases can help assess the plausibility of a suspect vehicle by comparing the distribution of cases to the distribution pattern of that food item, in consultation with regulatory and industry partners. For example, widespread outbreaks by definition are caused by widely distributed commercial products, and some foods are more likely to be distributed nationally, like leafy greens or packaged cereal, whereas others are more likely to be distributed regionally, like chicken or ice cream, or locally, like raw milk. Time characteristics, displayed by the shape and pattern of an epidemic curve, can provide clues about the shelf life of a product and/or the duration of harvest of a contaminated field. For example, cases spread over a longer time period may suggest a shelf-stable or frozen food item, ongoing contamination in a food processing plant, or other sustained mechanism of contamination. Conversely, cases with illness onset dates spread over a short period of time may suggest a perishable item, such as fresh produce. The third piece of information is case exposure assessments or case interviews. Rapidly collecting detailed information, including food histories, from cases in an outbreak is the most critical step in identifying common exposures. Communicable disease investigators usually conduct routine interviews before the case is found to be part of a cluster or outbreak. When investigators detect a cluster, the first step is to review the information contained in the routine interviews. If routine interviews do not produce any obvious hypotheses, investigators might consider conducting hypothesis-generating interviews using a hypothesis-generating questionnaire, or HGQ. These are longer questionnaires with extensive exposure history questions. Other methods to ascertain exposure history include reviewing consumer food purchase data, such as shopper cards. Generating hypotheses is the first and crucial step before investigators test hypotheses. Testing may include analytical studies or traceback investigations. Having a good hypothesis before testing will help narrow the focus of the investigation and make studies or tracebacks more efficient. Generating hypotheses is a critical step in any foodborne illness outbreak investigation. Having a good hypothesis can help narrow the focus of the investigation and make analytical studies or tracebacks more efficient, increasing the likelihood of rapidly and conclusively implicating the contaminated food vehicle. This video reviewed the 3 primary pieces of information needed for hypothesis generation: known sources of the pathogen causing illness, person, place, and time characteristics of ill persons, or descriptive data, and case exposure assessments, or case interviews. These provide vital information for hypothesis generation, particularly when used in combination and revisited as the outbreak progresses. Visit cofoodSafety.org for more information and tools to help with hypothesis generation.